I had a recording in a lab yesterday that, for whatever reason, didn't record. So not your lab. So hopefully it's not a problem inherent to the lab. We'll have to deal with that subsequently. And nevertheless, we'll return to where we were. So, so today it's interesting. Well, I'm going to pretty much work on the eye most of lecture today. Probably will get through it, assuming the degree of detail that's there. And we'll look at the anatomy. And if I have enough eyes upstairs, maybe we will dissect the eye this afternoon in the laboratory. And I'll fill you in the lab when we get to the lab of what's up with there. But the reality is, if you're not comfortable doing that, don't watch. There's always somebody bold who wants to do that type of stuff. But I was looking at your, they give us the list and what your majors are and things like that. So I mean, if you're, many of you are in the nuclear med track, correct? Yeah. Well, you're going to be sticking people. Having just been stuck in a CT scan in July, and it was competently done enough. So to a certain extent, you got to get used to doing that. It's very good. So all that lovely abdominal scan. Just tell everybody when you give them the contrast, it feels like you're a toaster oven. It's like, warms you right up. More on that. Are they still doing? Is radiation biology still in your nuclear med folks? Is that next semester?
I think it's next year.
Next year. Yeah, because it's Dr. Buxton and I. And did you have Dr. Buxton for physics for allied health? Okay. So Dr. Buxton and I we co-taught it on site 2 years ago. Whoever was supposed to teach it didn't stop coming in. So he got the radiation and I got the biology. And then after that, we did it strictly online. I don't know what they're going to do with it. I guess Dr. Monda, that's her bailiwick. That's whatever's going on. So more on that. We'll know more as the time comes. So I guess you have folks don't have to worry about that for another term. Okay. Very, very good. It didn't frighten you too much Tuesday, did I? Going too rapidly? Tell me. I don't get offended that easily. Believe me. My wife lets me have it if I do something wrong. Believe me. So I'm well past that stage. So if there's something you need, feel free. If you want to ask a question, ask a question. If it's not clear, I mean, I understand in the environment, it seems to be different in school today. You either have one person who's a constant questioner, and you know what I'm talking about, or a lot of times people just don't get as interactive. And I don't necessarily force interactions. I'm perfectly content. I can talk all day. That's what they pay me to do, is to talk. Rephrase. That's what you pay me to do. Thank you. Just saying. So this is kind of where we left it. We'll look more at the anatomy of it before. Okay. So at least I got a handful of smiles for a change. So again, just to reiterate the important points about all of these things, when you begin, and these are some of the, when we get into the lab, part of a typical lab practical will have this picture without the labels, and you'll have to identify 3 or 4 structures, okay? Not such a big deal. These are terms you should probably be familiar with already. You have the pupil, which is the opening. We'll talk about that today, why it opens, what's involved with that. The iris, the pigmented area, which is really an extension of pigmented, and obviously there's a variety of genetic factors. And I will qualify or disqualify myself right away. The eye is by no means my long suit. Okay, when we get to endocrine and blood and heart, blood vessels, those kinds of things, that's long suit material for me. So I'm trying. I wouldn't call this abbreviated, but I'll try to be at least somewhat limited in its degree of detail. Anatomically, it's fairly straightforward, and it's a very interesting dissection. The white of the eye is referred to as the sclera. Sclera is like when something is sclerotic, it's hard. Okay, and so this is a hard layer of connective tissue made out of collagen. And I'm just reiterating what I said at the very end last time. And it basically the way it's arranged, it has that white color. It's very protective. It really helps to hold the shape of the eyeball, as it were. And it varies when it gets overlying this area. Remember, I was telling you a little bit about cataracts. This is one of the areas. It's called the cornea, and the cornea is clear. Like if we do the eye dissection, you'll see with the obviously, these were eyes that were extracted from a sheep that died, and they utilized the body parts for anatomical specimens that are there. Those are the ones we typically use because they're the most cost friendly. And it clouds over, just the way if someone in repose, if you're going to a viewing, they'll have the eyes closed because it's pretty uncomfortable to see somebody with their eyes all clouded over. Though we often see if somebody has cataracts at who's still alive. And then that little reddish bit, that's the conjunctiva. That's a clear membrane, but it has filled with blood vessels that become irritated or enlarged from a variety of things. Trauma, think of a person with black eye, someone who gets struck in the eye. Allergies will do it. Alcohol, as I said, is a potent vasodilator. And so that's why you see like bloodshot areas in the nose when somebody's a long-term drinker. I should check my oldest and my youngest. My middle one's like me, doesn't drink. Okay. Accessory structures are the other ones. So eyebrows, eyelids. To me, the conjunctiva is not really an accessory structure. That's protective in nature. It's really important. And what happens, and maybe you explore, did you folks have to take micro? Okay. Maybe you explored it, maybe you didn't. But conjunctivitis in children, it's typically dirty hands rubbing. In adults, it can be from upper respiratory infections that spread into the area. And you'll see one of the regions, the nasolacrimal duct. Basically the tear duct. It can come from the nose and go up, and that can travel to the ear for ear infections. So it's very, very similar, globally speaking, germs, typically viral, but it spreads like wildfire. So if you want to have kids and you have, like we had 3 kids in a very short period of time, okay, when one gets it, they all get conjunctivitis because you're in such close quarters. Everybody's using eye drops. It's not minimal. The lacrimal apparatus is really the tear apparatus. I'll show you. There's a good illustration, and it's almost the analogy I like is sort of like maybe an unusual kind of like a windshield washer wiper arrangement that you have in your car. And then the extrinsic eye muscles. Did you do these when you did cranial nerves? Okay, lateral rectus, superior rectus, medial. Ringing a bell somewhat? Good. We're going to review them again because we're going to do the cranial nerves again. But I think that it's valuable. I'll try to get the cranial nerves in today because it's a fun thing I like to do. Okay. So supraorbital margin on the frontal bone is where you find the eyebrows. That's what they do. No big deal. Eyelids, they sometimes call them palpebrae. Again, I'm not going to ask you a bunch of questions about those. Where the commissures, the caruncle, where they came up with that name, I don't know. It's that bump that's in here. There's at the medial commissure contains oil and sweat glands, and this is just that anchor muscles and the things that lift your eyebrows up like that. No big deal. Eyelids blink a lot, as you can see here, and depending on and it really is just spreading things to keep the eye continually moist. And the eyelashes, they're reflexive in nature, they're protective in nature. There's glandular structures that produce the secretion. They're called tarsal glands. Not a big deal. If I say not a big deal, I'm not going to ask you a question. Okay, because my sense was that Tuesday I frightened you, not you. Dan, no. That I just was. Sometimes you tend to be. There's a very important saying: beginnings are such delicate times. So if you were a fan of the Dune movie, it actually comes from the Dune book that was written about 1958 by a guy named Frank Herbert. So if you like Zendaya and Timothy Chalamet in that movie, okay, which is an okay production of that particular movie. And that's where that comes from. So, and the ciliary gland, modified sweat gland. Okay, so there's a look at that clear area that we call the cornea, and you can see they sort of teased out to give you an idea of the sclera, but again, they are continuous. That's the outermost layer. You're going to see a recurring theme of layers. And depending on where in this case, in the eye, the functional layer is inside your retina, where you're able to see. In a lot of tissues, it's not. Sometimes the functional layer in your gut is the inner layer, and in your arteries and veins is probably the middle layer, or even the outermost layer. So it depends. Those kinds of things. So there's a lot of stuff here. I don't go crazy. Okay, you can get little bumps. It's got a fancy name. I probably couldn't pronounce it. Styes, we used to call them that a lot. It's just one of the glands gets inflamed, generally a buried eyelash, drops, ointments, whatever they're using. And the same thing with conjunctivitis. Okay, so they were things that were done. We use different remedies today. When I was a kid, they had things like that. They used mercuric oxide yellow. You didn't need a prescription for it, but we didn't go to the doctor very frequently because there wasn't health insurance. And mom, they used a lot of home remedies for things like that. And so you had to pay a substantial amount to go to the doctor. It was a totally different time. So I grew up, I was born in 19, if you're curious, hopefully you're not. I was born in 1951, which I wager I'm the oldest one here. Okay. I think. Probably correct. So the conjunctiva, transparent membrane, and there's different parts of it where it underlies. You're going to see more of that when you look at the heart and the lungs. These are layers that are sometimes they're against the eye, in the enclosure, and sometimes against the organ itself. And there's the bloodshot thing and the sac and all that good stuff. Conjunctivitis, why do we call it pink eye? Because when the blood vessels engorge with blood against the white background, pink eye. Sometimes it's more severe than others. So it is. I'm reiterating the point, but it's pretty significant and it's very and contagious. It's just the word. The tear apparatus is interesting. It kind of goes from upper outer, and the tear duct is sort of lower inner. So imagine, like one of the vehicles I had that we hit it in the back, and it came down, and then the wipers went. This is kind of what happens here. And as you can see, the lacrimal secretions, the liquid, is mostly a salt, but dilute solution, because if it was very salty, it would certainly irritate things. Mucus, antibodies, it's supposed to be really a favorite area with minimal microorganisms. Okay, and you'll see kind of the way it goes. So it's a pretty neat diagram. I wouldn't use this on an exam, to tell you the truth, but you can see up here, there's the apparatus, and so as it secretes, and as you blink, or you open and close your eyes, it kind of distributes throughout. And then the excess is picked up there. So it's a pretty neat anatomical arrangement, I think. Nasal cavities, so I was telling you before, cold, sinus infections can all cause problems in it. A lot of times when you get a cold and your eyes start to water, or maybe if you're like me and you have allergies, particularly this time of year, those are the kinds of things that you'll notice. You said you had these before. A quick review, never hurt anything. Because they're the easiest thing. If you're going to have a test, I have the solution. Okay, it's a never forget. They taught us this in medical school, these 50 years ago. Okay, so I don't know if they shared this formula with you. LR6 SO4 3. Commit that to memory. Or like when you have a test, you write it down right away. Okay, is that ringing a bell? Huh. I feel. So here's not a great artist. Eye, nose, muscle, 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 muscle. There's 6, and then there's oblique muscles here and here. So these are straight muscles with straight. We call them rectus. So superior rectus, because this is at an angle. Superior oblique. You'll see the illustration. Medial rectus, I'll just do MR. Lateral rectus, inferior rectus, inferior oblique. And basically they're the ones, so when you're able to, if you see somebody, doing a neurological survey, follow my finger, that type of thing, move it up, move it down, following the flight of something, we do all the time. Those are the muscles that do it. They're called the extraocular muscles. They're innervated by 3 cranial nerves. And 2 of those nerves is their sole responsibility. That's the only thing they really do. The lateral rectus is the sixth cranial nerve. You'll learn about it a little later today in lab. That's called the abducens nerve. The superior oblique is the fourth cranial nerve. I don't care whether classically it's Roman numerals, that's why I'm doing it that way, but you can use regular numbers. I don't care. The test, I don't care. I'm happy there. If you're close. Cowboys hat.
No, it's a round horse.
Oh, thank God.
No, not Cowboys.
I don't know. My daughter-in-law is wonderful. Did I tell you she's a New England fan? They're from Massachusetts. Yes.
Could you explain that one more time?
I sure could. Lateral rectus, this muscle, is the one that lets you move this eye out. So if I'm gazing that way with this eye, the lateral rectus is pulling out. And it's the sixth cranial nerve. We're going to see it in the lab. The superior oblique kind of rotates your eye because of the angle sort of downward. It's coming in like this way and sort of like you're gazing sort of down and sideways. That's the fourth cranial nerve. Its fancy name is called a trochlear nerve. It's not a big deal to remember it. Okay. And the three at the end, all the other ones, other than the lateral rectus and the superior oblique, the superior rectus, medial rectus, and inferior oblique are all innervated by the third cranial nerve, which you'll come to know as the oculomotor. That's a really important nerve, and we're going to get to it shortly. Because that's the nerve that opens and closes your pupil. It's the old story. If I turn the lights out in here, and we were going to watch something, I want the lights out, and all of a sudden turn them on, it's usually a good idea to give the students warning because it's kind of like, ugh, right? That's the one that adjusts the pupil with regard to light reaction. So that's really important, and it's a really important topic. That's there. It's one of the things, if you're going to be in a field where you interact with people, those are little things you look for. Okay, if you were looking at somebody, and I'll speak to the nuclear medicine students. So maybe you're doing a brain scan for some reason. Well, you might be observed an irregularity in the pupils or some sluggishness as they begin to look. That's typically a sign of concussion. So there's little, and they're great for everyday life. Like, yeah, it happens all the time. If you have kids, they get clunked in the head all the time. They just do, depending on what sport they're doing. And most of the kids do sports. My boys were hockey players up until they were like 9th or 10th grade. My daughter was a figure skater up until that same age. So you're always falling on the ice, clunking yourself. So LR6, lateral rectus 6, superior oblique 4, all the rest 3. Happy dance. Done. Moving on. So they originate from the orbit, the bony structures around there. They help to follow moving objects. The 4 rectus muscles happen to come really from a ring of tissue, not a big deal. They're their names. The oblique, and you can see it rotates the eyes, like I said, downward and laterally. So it's like taking it because of the angle, it kind of rotates. You'll see the pictures in a second. So here's what it looks like. Here's this ring structure they're coming from. So you begin to see this within the structure of the orbit. This is a little bit easier, I think, when you begin to see it this way. So here you can see it's like this is the superior oblique. When it fires and pulls, it kind of is pulling the eye out that way, and because it's on the top, pulling it down is a way to kind of think about it. I'm not going to ask you about the actions. I'm just trying to give you an idea what the relevance is, and it's the type of thing that we look for. And when they're not coordinated together, that's when we often hear about people having something called cross-eye, and there's varying degrees about that. My middle one we noticed it when he was 3, it's like a lazy eye. And we took him to an eye specialist in Pittsburgh, who was working on children's, and basically said, do we have to do something? No. Okay. Because there's so many degrees of it, this wouldn't affect him in any of his activities whatsoever. And he drives professionally, he drives a truck. You get a commercial driver's license. Okay, so here's how they look. And so this is the same thing. All the rest 3, those are the oculomotor, LR6, SO4. Business done. And this is what happens when they're not coordinated. You can't focus necessarily, results from paralysis, weakness, neurological disorders, And surely the most common of these, if you think in a traumatic sense, would be a concussion sometimes. And here's the cross eye where it's congenitally weak. It rotates the eye in one direction. Brain begins to sometimes disregard it. So, and your body does this. Something's sending mixed signals that sometimes you tend to ignore. It's an interesting phenomenon in and of itself. That's everything of that stuff, okay? Now we're going to get to sort of the components of the eye themselves. Again, the recurrent theme of this discussion is layers, okay? So you're going to see this repetitively, whether we're in reproductive, whether we're in gastrointestinal, you're going to see multiple layers. In the case of the eye, the outer layer, and you will see on the dissection, they're all three layers are easily visible. Okay, the outermost layer is fibrous. It's hard. That has two items in it that are really continuous with one another. As you will see, the sclera, the white of the eye, which occupies about probably 80 to 90% of it, and that little bit of cornea that's clear that strictly overlaps the iris and the pupil. The vascular layer is really important. Another name you'll see for vascular layer is this, and you'll see it shortly. It's called choroid. There's some overlap. It has a great deal of pigment and as well a great deal of circulation. And so the pigment comes from the structure as well as from the circulation. Effectively, what you will see, for the easiest way to describe it, it acts like a dark room. I realize you are the generation of, and as am I, of digital photography. We used to use film. Just ringing a bell? See, I like that. Thank goodness I thought it was a Cowboys hat. I was gonna have a hernia. Okay. So understand that years ago you would get a camera, you get a film cartridge or a roll of film, you put it in there, and then you'd have to very carefully take it out. And effectively, that's where we would develop it. When I used to take X-rays in my office, we would dip them, convert a closet with a red light, to a dark room, and basically hung them on hangers and dipped them in a series of solutions to develop the X-rays. X-rays are effectively negatives. Okay, now everything is digital. Okay, it's the reason why Kodak, which was such a giant company, they didn't think digital photography was going to catch. They thought it would always be film. Oops. That cost thousands upon thousands of jobs in Rochester, New York. Why do I know that? My sister and her husband lived in Rochester, New York for more than 40 years until they retired. And she worked for a big company there called Bausch & Lomb. But Kodak was immense. Okay. So look at the vascular layer and some interesting nuances. The continuation of that vascular layer makes the iris. Modifications in that vascular layer make up something that's part of your parasympathetic and sympathetic nervous systems. Part of your autonomic nervous system is called the ciliary body. That plays a pivotal role in shaping your lens and opening and closing the pupil. You'll see more about that. And then lastly, the inner layer is the retina. This is the functionality. This is where you see from. It's a very interesting layer. It's very, very delicate. When you open the eye, dissecting wise, it's very easy to lose it. It's a thing. It looks for all the world just like a piece of fatty membrane. And a lot of times when students are doing it, and it's not necessarily them, a lot of times when the eyes are extracted for dissection, they just lop them out traumatically, and it messes them up, and it effectively, quote, detaches a retina, which is something that happens in people when they get older, something from injuries, etc. What holds this retina, this delicate structure, against the pigmented layer, so the scatter lets you see a clear picture, and against that firm outer border of the fibrous layer, okay, is a very large sac of a clear gel-like liquid we are born with. It's called vitreous humor. The old term for body fluids was humors. Okay, so when they said, and if you're like me and like all this historical stuff, You see those old, like the old movies and things like that where they depict ancient and they bleed people. Yeah, because they didn't know how to treat them. They didn't know anything about antibiotics, things like that. They would bleed people to try to make them better. They had the gout, they bleed it, and because you had too much of a certain fluid. So there was phlegm, there was bile, there was black bile, and there was blood. So all that's if you're interested in the medical history of that, that's where it came from. So they were four body humors. So we still utilize the term because it's older. Nervous system, we see older terms pretty consistently. So you have vitreous humor, and in the front you have another humor that's more liquid called the aqueous. The name would suggest it's more water-based, not gel-like. And the separation between the two is the lens, and it divides the eye into two compartments. The anterior compartment, very small, and the posterior portion, quite large. You can see here, compartment, segment, whatever you want to call it. Voila. You'll see this illustration again because it has the sclera, and notice how it changes here, even though it's the same thing, into the conjunctiva. Here's this pigmented layer, another name for it again, choroid. And as you can see, as the choroid goes anteriorly, you begin to see it develop what's called the ciliary body and these suspensory ligaments. They're strands of this pigmented material, and you frequently see this on the dissection, that attach to this rather dense structure called the lens. And it's that lens in combination with the cornea that allow us to focus our vision appropriately. And indeed, if you wear, like I do, glasses, or if you have contacts, whatever, or if you've had some type of surgical procedure to fix it, basically, you're trying to make sure that the impulses go in the right direction. Okay? And the whole idea, the ball game, so to speak, is to go here. More on that in a second. So you have the suspensory ligaments, and then you have the continuation of that pigmented layer. That's the iris. But within the iris is an opening that can open and close, and that's the pupil. So effectively, that whole middle layer, the vascular layer, is responsible for this, really, the development of this ciliary body, which is under autonomic control. We aren't involved with it, so to speak. We don't have conscious control over it, as it were. Positioning of the lens, as you will see shortly. So if you're looking at the board from where you are, your lens, even with or without corrective lenses, the lens in your eye is relatively flat. It's designed for distant vision. When you're working on your computer, when you're looking at your phone, that's close vision. Your lens gets a lot rounder. Okay, and it all has to do, and this is my weak point, because it involves physics, and I don't do physics. I don't do plants, and I don't do physics, and I don't do teeth. The whole idea is to focus the light, depending whether you're looking far away in the distance, like when you're driving, or you're doing close work, here at the point of maximum vision. And it's called the macula lutea, and there's a little indentation in it called the fovea centralis. And what's interesting about that is it doesn't go straight back, because that's the blind spot. This is it's a purely sensory nerve. So in yellow, you can see the retina. And what it does, it's basically, think of the nerve, even though it's bringing the signal in, if you think of the nerve, quote unquote, entering, it spreads out along this retina, all the cells that make up this elaborate vision structure that we see with, and they spread out. So all the nerve endings, technically where you and I see things, okay, are being all fed back by a common exit point, and it's called the blind spot because there's no sensory apparatus there. Moving on. And as you can see, it's arranged kind of interestingly. Normally, blood vessels are on the outside. In this case, they're right in the middle of it. And there's like a pretty significant stump that you'll see on most of the eyes where the optic nerve exits. So, layer by layer. Outermost layer, dense avascular connective tissue. Thick. If it's damaged, it's going to scar. It's well protected. There's a lot of fatty material in the orbit that surrounds it. The sclera and cornea, the sclera opaque, hence the white, protects in shape, place for the eye muscles to attach. Where the optic nerve exit, the sclera effectively becomes, if we go backwards, this becomes continuous. Maybe it's a name you remember from meninges or meninges, the dura mater, which means heart material, dura for durable. The cornea, transparent, only about a sixth, so 15%, let's say. It's a window, best way to describe it, bends the light. The light bends 3 times. As it comes in the cornea, which is convex, bends here. When it comes into the lens, which is convex and then concave, bends twice more, from what they tell me. Covering on it, specialized lining materials. As you can see, there's what they call sodium pumps. They maintain the clarity. So when somebody dies, it's a pump, it doesn't work because it requires ATP and they're not making it anymore. Okay, a lot of pain receptors that are there. You know, it's like we get something in your eye, it's like, oh my God, right? Vascular layer. I never called it the uvea because I don't know anything. Choroid. Blood to all layers. It's a blood supply. The pigment is also there, so it avoids scattering of light. Just the way if you take a digital photograph and you know, particularly if you're trying to do it. We do it all the time because we sell stuff on Amazon and eBay. My wife and I do, and you got to take good pictures. And so you're always finagling with the light. So in the anterior, the forward portion, it becomes a ciliary body, thickened. It's got bundles of smooth muscle. They play a role, as you will see. They're all part of it. Blood vessels. And then this, what they call the zonial. A better name is a suspensory ligament because it holds on to the lens and it exerts a tug. And now we're going to get to the important part. Okay, the iris is the continuation. The pupil is the opening. So what happens? Closed vision, like this, bright light, these muscles are sphincteric. If you remember from muscles, a sphincteric muscle, remember you had the orbicularis oculi and the orbicularis oris. They're ringing a bell. Close your eye. Sphincteric or circular muscles kind of roll this way. And when it does this, those circular muscles, they'll contract the pupil. It's interesting what they do. They actually release, and you'll see the illustration, the tension on those suspensory ligaments and cause the lens to get rounder. When you, like if you're looking distant, if I'm looking over at the main building over there, all of a sudden my pupils will open, okay, for distant vision, driving in a car, whatever. And at that point, when it opens, it actually creates a tug to straighten out. And you'll see that in a second. It shows it better than I can explain it. So distant vision and dim light dilated. So, and when we look at it in a sense of doing an examination, you have a little light. Everybody carries around a pen light, nurse, doctor. And in a relatively darkened room, you do pupillary light reflexes. So you have them look like this. They bring the light from the side, okay? And theoretically, assuming that there's no concussion, as the light comes there, it'll cause the pupils to close at the same rate, whether it comes from this side or that side, because you test them both. It's called the pupillary light reflex. It should be equal on both sides, not sluggish on one. And when you get to know what's fast and slow, okay? And anytime you see asymmetry, where one is fast and one's slow, so that's the pupillary light reflex. At the same time, okay, we also test for accommodation. So you might remember having this test if you had your eyes checked, I'm sure you did. You know, they'll say, follow this. They'll take like a pencil and you'll do this. Your pupils will not only constrict, get smaller, but they'll go medially as you're following that as it gets closer, let's say, to your nose. That's called accommodation. So we use an acronym for it on a medical chart called PERLA. Pupils are equally reactive to light and accommodating. And all of that, everybody, is testing that third cranial nerve, which we'll go over again in lab, the oculomotor. It's one of the 4 cranial nerves that has parasympathetic, read that as glandular activity. That's a big player. So what they used to do in the football games, Now they have that blue tent there because it's easier to examine somebody, and I think offers a certain degree of privacy, particularly if it's significant injury. Okay, but you used to do that in the sideline. That's what really those cranial nerves are about when you do a neurological survey. And there are different things. Emotional states can do it, et cetera, et cetera. Same illustration. So here, this is what I'm showing you. When it's parasympathetic, when you're comfortable, you're reading, you're looking at a book, you're studying something, you're on your computer. That's a relatively relaxed state. As you can see, it constricts the pupils, okay? And when the sympathetic, remember fight or flight, you want your eyes wide open so you can see where the danger is, who's chasing you, where that car is coming at you so you can respond, etc. And because of that sphincteric motion, and they'll show it to you later in some illustrations, the retina is really develops in utero early. It's an outpouching of the brain, so that cranial nerve starts to go, and it's got. And ladies and gentlemen, you have, I mean, surely more than half, and I think that's conservative, of our nervous system is dedicated to vision. So there's a ton. They're called photoreceptor. They receive light. Okay. So you have photoreceptors, you have nerves that support it, and you have connective tissue. And you might remember the name for connective tissue we call them glial cells. The longer name is called neuroglia. They're astrocytes and oligodendrocytes and Schwann cells. Are those names ringing a bell? Yeah, that's so those are part of the deal that are there. And it's very delicate. It has that layer, this retina, because it's very, very thin. The outer layer has a certain amount of pigment. The inner layer is really the nerve itself. And interestingly, the light kind of, should have done it that way. The light kind of comes in and it goes all the way through the retina to that pigmented layer. That's where it triggers vision. It's not so much on the outside. It actually goes through that layer, triggers vision, and then comes back in this way. It's kind of interesting. And it's the reverse of the way most nerves work. Okay? Most nerves, okay, I'm thirsty. I'm going to get a drink. So my mind tells, get this, open this, have a drink, right? So you're activating the system. When you're vision, it's the opposite. I'm not creating an action potential. I'm stopping one. It's what's called hyperpolarization. You might remember the terms depolarization. When we fire something, hyperpolarization, it's like I wanted to be this morning, not come in. I was tempted. You guys probably wouldn't have been upset. Nah, go ahead. Come on. Spent a lot of time as a student. So all that, going too fast. So delicate layer, you're going to see sort of the internal structure. So the light goes in through the cornea, bends again, outer lens, bends again, inner lens. So it's convex, concave. Directed here. That's that best area, the macula lutea, the clear area with an indentation called the central indentation, fovea centralis. So here is the optic disc or blind spot, which you can see when they focus on that. So the pigmented layer, a single cell thick against the choroid, so it's deep comparatively. The light's going through it to that pigmented layer. All the way to the ciliary body, absorbs light, prevents it from scattering, and it begins to work on something called the photoreceptor fragments. You're going to see that a little bit soon. You're constantly making new photoreceptors. We make them. Once we use them, they immediately break down. And we make them again. It's continuous. And they also store vitamin A. Vitamin A is a big player. Do you have to do in your field's nutrition? Is that in the package of stuff that you have to have? Yeah, I don't know. Okay. It's interesting. The fat soluble vitamins are A, D, E, and K. We tend to people are worried about them because we store them so you can take too much or not have enough. B vitamins are pretty easily go through you. Yeah, and it's easy from what we are. Vitamin A is something that you can get from diet. Some people have problems with it. There are some problems with excessive amounts. I'll talk more about vitamin D, which I think most of us have not enough of. I think vitamin D is a really important thing. But just sort of throwing that out there to start. Vitamin A, it's carotene. You get carrots. Your vision is better. So the neural layer is transparent. Okay. And you're going to see it has a serrated edge. You actually see that on the dissection very clearly and distinctly, called the ora serrata. Okay. And you're going to see the cells. There are 3 cells. And now we get to what makes this a special sense. You have the pigmented cells that change shape with light. They're called photoreceptors. There's 2 kinds, rods and cones. Terms I'll bet you've heard. And you can never forget cones, CO, for color. You can't get that question wrong. Cone is the name CONES, and they're conically shaped. They've got like it's a sugar cone, not a cake cone, in ice cream terms.
What is it?
Wait, I'm coming.
Oh.
Pardon me?
Go ahead.
Yeah. Do you know the difference between a sugar cone and a cake cone? Like when you get soft serve, it's usually a cake cone. But the old style sugar cone, like the ones that kind of get in from the bottom, kind of suck the stuff out. I have an existential relationship with food. I eat, therefore I am. We made Asian style beef, broccoli, and cashews last night. Goodies. Fresh ginger, garlic, green onions, flavor base, oyster sauce. Food. So, yes, ma'am.
What did cone stand for?
CONE. It's the receptor that lets you receive color vision, and the rods are the receptors, and they're based on their shape, which you will see shortly. And the rods, it looks like a rod.
Wait, say it again one more time.
Okay, cones for color vision.
Color vision.
And you have about 20 times more rods, which think of black and white. It's like an old movie. It's like Wizard of Oz starts in black and white, color once we get to Oz. Come on, you also have. I'm a big Wizard of Oz fan. Like, wait till we get to the heart. We dissect. I play If You Only Had a Heart. You heard that song? Tin Man? But you don't believe me. So here they are. The optic disc, no photoreceptors, no name is a blind spot. Retina. In each eye, 250 million photoreceptors. Billion with a B. Rods and cones. Okay? Same illustration. Rods. Dim light at night, shadow, shade, silhouettes. Rods don't require a lot of light to be signaled. A lot of us have different degrees of night vision. Some people go outside at night and a little bit of light they see very, very well. Some folks like me have a hard time seeing at night. Not if I'm driving a car with headlights on, but other than that. And so they don't require a lot of light. There's about 20 times more of those than cones. Okay. No sharp images. It's very indistinct. So, and they're concentrated more on the outer areas of the eye, what we call the periphery. Okay. And they'll test for that with vision. They'll bring like a finger in this way for visual fields. Yeah. So some folks have an extensive amount of those genetically. And it really isn't advantageous. So if in the military, that would be advantageous for things like that, for certain fields where you're working a lot out at night that are there. So it's a whole lot more. They don't require a lot of light. So it's a lot of, think of it this way. So around here, we're getting to that wonderful time of year, deer everywhere. So how many times when you see a deer on the road in front of you, it's like it's magically there. It's right in front of you. And it's like, oh my God. Versus how often do you see an object moving along the sides kind of alert you to a problem that are there? We typically see things more peripherally with our peripheral vision. So it gives us like a hint to respond to it, particularly at night or at maybe twilight, that type of thing. In my neighborhood, they're everywhere. So cones, high resolution, gotta have light. Just like taking a really good picture, the lighting has to be there. It's there. The area where we focus this is called the macula lutea. It's just alongside of the blind spot. It's nearly all cones. And the tiny pit called the central pit, fovea centralis, is effectively all cones. Okay. And so we tend, and a lot of times we'll see something and we'll change the way we're looking at something just so it will concentrate it in that area so we can see it better. We've all done, and this is a very telling, it's a very good illustration. Another one of those, you'll see me again. And because despite what you may have heard, I'm nice. I will even give you a list by number of the illustration. You know, but I didn't say that. I'll stop recording. See, I can be crazy because I can tell I'm having Alzheimer's. Oh, I'll do that. Cones. Notice the flat end. Okay, we have bigger part rods. Notice the flat end. Cones. Many more rods, relatively few cones, despite not as many as this illustration. Here, the light comes in this way. This is that pigmented area of the retina. Okay, a lot of this is gobbling up pigments that are we're constantly, like I said, breaking these down and recreating. Every time they're triggered, they break down. They change shape and they break down. So you get that. Now, what makes special senses special, I'm in class. Is this. Okay? Here, these are called photoreceptors. So you're changing light to an electrical signal. Now we have to convert that signal in some way, shape, or form to something in the brain. So we have another specialized sensory nerve. You learned about these, they're called bipolar, sensory cell. They're called bipolar. So really all they are is getting a signal here, kind of doing something with it and bringing that signal along to the next layer of cells called ganglionic cells. So you have the photoreceptor cells, and you see bipolar cells in the nose for smell. You'll see them in other areas for taste. The only place where we really see these bipolar cells are special senses, because we're constantly transducing the signal, whether it's a pressure or chemical or a photo signal. Now we have to convert it to some form our brain will understand. And it's collected by these ganglionic cells because they can connect multiple ones, and they pass them along into the nerve itself, which is a sensory nerve, going back to your brain, effectively. And you'll see the pathway momentarily. Just tell them. Messages. So, and here's what it looks like. Here's that choroid, the pigmented layer. Here you can see the retina's own pigmented layer. Here you can see here's the, like I said, here's the choroid, here's the retina's pigmented layer. These are the rods and cones. And then you have bipolar cells. Nuclei typically stain purplish, blue or purple, as you can see here. So it's kind of the same thing, but the schematic is a little bit easier to understand. So the blood comes from the choroid vessels outer third of the photoreceptors come from the choroid. There's an artery and vein inside for the deeper portion, the ones that are kind of heading back eventually, okay, and by way of the optic nerves, and we can see them. That's the beauty of that. It's the eye is an interesting area. So if you've had your eyes examined recently, now they tend to do all of these tests at once. They have a device if you go to an eye doctor, and it will measure pressures. It will take a picture of the blood vessels. Years ago, they'd have a device called an ophthalmoscope, and it took some skill to look in the eye. And very often you had to have your eye dilated. Okay, so they put drops in your eye because it make it easier to look inside, and then and because it stayed dilated for an hour or so, light bothered you. They put these these like paper or plastic sunglasses on, and you had to bring somebody with you because you wouldn't drive. Now they don't do it so much because now they have this device. If you go to any eye care center, you're going to see that. So that's part. And that's what these are. So what's interesting about it is these are arterioles, tiny arteries. Now, an arteriole in your eye, an arteriole in your kidney, an arteriole in your hand or your foot all look the same. Doesn't matter. They're all the same size blood vessel. What happens in one, exclusive of some unusual condition or some genetic condition, happens to all of them. And where the other place you have arterioles is your heart, a lot of them. But you can't really look in your heart, but this gives you a snapshot. So if you look and see this, okay, a nice straight area like this without any little nicks or irregularities or areas where it's not continuous, where it looks like it narrows, that tells you the artery, the blood's flowing nicely through it. And so that's one of the ways a doctor or a nurse can check inside and see that there's adequate circulation. It's a real good test. People who smoke, often those are things that are affected. And it's a quick way to look, because if they're narrow here, they're surely narrow in your heart. The good news is if you've damaged them because smoking, alcohol, whatever, pick a bad habit, all those types of things, if you correct it, it tends to correct. You have time. Look, if the damage is done, then it's permanent. You can lead to blindness conceivably. That's why it's such an important part of evaluation. Here's the detached retina, okay, where it separates, and that jelly-like vitreous, which you'll see, kind of seeps in between them. That can be a big problem. And usually trauma, I guess bungee jumping would be an example, but it could be any kind of trauma. We're a little concerned about my wife's mom, who's 97 plus right now. And you get they're getting seeing floaters or little flashes. And she was just 97. My wife was very funny this morning. She goes, when you're that age, you don't even buy green bananas. You never know. And she's not being flip about it. We had her birthday dinner August 20th, which was a Sunday. In attendance were her 101-year-old brother and her 92-year-old sister. So she could be here for a long time. And that's what that flash floaters, and there's ways to remedy that if that's the choice. Here are the chambers that you're seeing. Okay, the posterior. And the lens is the divider. When you look inside that eye, you'll be very impressed by the size of the lens. The lens will be clouded over, not clear, for the same reason as the cornea be clouded over, because the mechanism to clear it no longer works because somebody's dead. There's no energy transformation. And that lens effectively divides these two segments. So the posterior segment has this vitreous humor. You're born with it. Transmits the light because it's clear. Pushes, it helps to really hold the retina flat. And it's part of when they measure something called intraocular pressure. Okay? Forms as an embryo, lasts your lifetime. If forensically, when somebody dies, you can sample that to try to get an idea, because it'll be reflective of maybe something that might have contributed to somebody's death. If you have an interest in forensics, something like that. The anterior segment is divided, okay, between the cornea and the iris, and between the iris and the lens. So it's very small, but there's not a lot to it. Okay. Okay. And the entire segment is called aqueous humor because this fluid is continuously formed and continuously drains out. So you're constantly making it and draining it out to a tiny canal inside the eye. It's got a great name, canal of Schlemm, nutrients, oxygen, et cetera, et cetera, et cetera, waste removal. And they should go through this whole thing with this here. So here's the lens, here's the vitreous, here's the eye. So there's a little bit of the compartment here and more of the compartment here, but they're all part of the aqueous. Why it is so darn important is that we make it at a steady rate. If we are making it too fast, you get too much pressure. If it's not draining rapidly enough, you get too much pressure. It has a very famous name for the condition where there's too much pressure, and they test for it all the time, glaucoma. And glaucoma can lead to blindness. Glaucoma is very painful. More people have it than you would think. You can be treated, okay, but it has to be identified. So, condition in which drainage of aqueous humor is blocked. Typically, it's blockage of drainage, but overproduction can do it. Pressure with the eye, dangerous levels, retina, eye, because it's pressing on the system. Blindness. All these types of things are there. And typically, unless there's a really strong family history or some genetic basis that would surprise you, or whoever's investigating it, typically about age 40. And there's, as you can see, there's treatments. The lens, okay, so biconvex means curved this way, so it looks like an ellipse. Boom, boom. Changing shape, has lining on it, has fibers. The fibers are this transparent protein called crystalline, okay? So the lens and it's constantly being added. So as we get older, the lens isn't as flexible. We're able to change shape. So people, when they get, I've been wearing them for 20 years or more, bifocals. Okay. Now you don't see, in the old days, you used to see the bifocals. Okay. Nowadays, they incorporate that into the eyeglasses. So these are all things. I told you before about cataracts, aging, okay, diabetes, smoking, people out in the sun a lot. And there's certain things are in. Some are there congenitally. Proteins become distorted. Excessive levels of vitamin C. You don't see a lot of that. Vitamin C has always been recommended for wound healing. A very, very long time ago, arguably the most famous scientist in the world after Einstein died. Okay. Dr. Linus Pauling, a two-time Nobel laureate. The cartoon Snoopy named the character Linus after him. Two-time Nobel laureate. And he wrote an article when I was in college. I haven't had a cold in 5 years, and I take megadoses of vitamin C. And everybody started taking vitamin C like it was candy. But the trouble is, there's going to be problems with it. But because he said it, everybody did it. It's an absolutely true story. And nowadays, it's miraculous what they can do with surgery, with eye surgery. It just may surprise. Nowadays, you can go in and have your cataracts repaired. You will come home the same day. Your instructions will be to not lift anything more than 10 pounds, and you're going to be using eye drops. You come back the next day. Absolutely. Within a few days, my mom had it done. Didn't have to use her glasses nearly as much, anything along those lines. Years ago, when they did that, 60s, even into the 70s, they'd sandbag people in bed for 10 days. Flat in bed. Couldn't move. To give you an idea of the transition in the surgical aspects. And that's that. So we're almost done with all of this. I have a little bit more to share with you. I know it's a lot. It's coming through. You'll be happy to know the first part of this doesn't count. I don't do physics. If you had Dr. Buxton, he handles the physics. He and I are at odds all the time because he's a Liverpool fan and I'm a Manchester United. We don't like each other. Okay. So visible light. So you'll see it and this is all the physics behind it. Effectively, the rods, as you can see, we really only see three shades, three colors, and mix them together: blue, green, and red. And from all of those colors, we're able to generate this vast array of colors that, just looking outside, trees, etc., all that type of stuff. The rods, and you don't have to know any of those numbers. So by refracting, due to the change in speed of the light when it passes from one medium to another, okay, and you saw this if you used the microscope, how it, you have to focus it, or you have to use oil immersion and micro. You're trying to compensate for this. So lenses of the eyes can also refract light because they're curved on both sides. Okay, as you can see, thicker and convex, thicker in the center, and the edges concave, thicker at the edges, convex, concave, indented. So, and it just changes the formation. And you can see effectively without me going into it, and I'm not going to. But this is the refraction, the bending, better way to say it. Enters the cornea, enters the lens, leaves the lens. The majority of the refracting power is the cornea. Okay? But it's constant in nature. So we either corrective lenses, glasses, or alterations in the lens itself, in which people are having lens implants and various kinds of surgical intervention. And this is the idea. Our eyes typically look distantly. We're not so much for close. So our lenses, and that's where you hear 20/20. It's the idea, once you get to 20 feet, you should be able to see a certain height at 20 feet. Anything past 20 feet, your lens really don't change. Just don't. Okay. Ciliary muscles are completely relaxed at that distance. Okay. So ciliary muscles, when they're relaxed, causes a pull at that ciliary zone and the suspensory ligaments, and they show it to you here. So here you can see what it kind of looks like. Here are those suspensory ligaments. Here's the lens. So the lens flattened. See how flat it is? So when the ciliary muscle relaxes, this is effectively what happens. And when it's relaxed, it actually goes into a more stretched out position, causing the lens to flatten and elongate. And we see things upside down and backwards. Okay. And here's the idea of accommodation. So as it's coming closer to you, nearest point, and we lose a lot of that as we get older. So you hear terms like presbyopia, okay, where there's alterations in your vision as you get. A lot of folks have this. You're very fortunate if you're able to see the longer you're around. And that's the convergence when your eyes are coming together. And here you can see parasympathetic. So I'm doing close vision like that, contracts the ciliary muscles, but so even though it's helping to contract the pupil, it's effectively relaxing this area here and allowing that to bulge. Okay? It loosens the zonule, it loosens those suspensory ligaments. So when you hear the terms myopic, they're nearsighted. Okay? I'm nearsighted. It's probably much more common than farsightedness, which is hyperopia. Okay? And so you correct them with different types of lenses. You don't have to know it, again, it's more physics. And people often say, I was told this, you have astigmatism. Little did I know there was an A in front of it. It's not a new word, stigmatism, it's astigmatism. And it has curvatures, and obviously can correct it with lenses. Then we hear about LASIK procedures. It's trying to make the eye rounded appropriately. And with the science today, you can do that, from what I understand. Okay? And that brings us to this. Let me see. We're gonna have to continue this up in the lab. Okay. Those rods and cones, okay, it's that segment that's the farthest away from the center of the eye, and that has these pigments. So I'll just give it to you in summary form and be a little more detailed when we start lab, okay? And so there's a lot that's involved with it. So here's what they look like in close-up. Here, the pigmented layer. Here are some additional pigment that's part of the retina. You can see the conical tip versus the rod. And you can see it's like a series of discs. They're constantly, again, being produced. And when they're done, after they're used, effectively you gobble them up. So All of that's part of it. They're embedded in those discs. And if you go back, you can even see the discs are there in the cone. It's just a different pigment with a different name. And as you can see there at the end, the most important part, light changes their shape. This is interesting. It's a photoreceptor, but what the light is effectively doing, it is changing the shape of those pigments. And they go from effectively a bent shape to a straight shape. And in doing so, that changes everything. So they're very vulnerable to damage. Retina's detached, really super bright light, like a flash or an explosion that you weren't prepared for. You'll lose that. Okay. But the outer segments every 24 hours are recreated. So what the treatment has been around for a long time. If somebody has one of those flashes and they're temporarily blind, they basically cover their eyes as completely as possible for at least 24 to 48 hours. So that absolutely no light gets in it. And it maximally gives the opportunity for the damaged pieces to be gobbled up and recycled and new ones to be made. It's been around for a very, very long time, that treatment. So a lot of times there's a reason for optimism if the damage is not so severe. Rods, very light sensitive, where cones are not. Okay. So, and they're not as distinct. When you have like 100 rods into one ganglion, that means you're getting a multiple signal. So you're really not getting, and where cones, it's almost 1-to-1. Like when a cone sees something, you get it. That's why you're so distinct when you see, and today with high definition TVs and upscale computer screens, it's remarkable. React more quickly. One of 3 pigments we talked about. Okay, detailed, highly resolution vision. Very, very. It's just like think of pixels on a camera or on a TV set. Okay, so brain can put together an accurate, high acuity or accuracy resolution of images. Summarized here: non-color versus color, high versus low sensitivity, so you need a lot of light. High acuity, pretty much 1-to-1, 20-to-1, rods to cones, peripheral, central. Plenty for today. So we'll conclude there and see everybody up at 2 o'clock in lab. How's that? And hopefully this recorded.